Hello everyone, it's my pleasure to introduce dual role of ramR mutation in enhancing immune activation and elevating eravacycline resistance in Klebsiella pneumoniae. Eravacycline, ERV, a novel antimicrobial, is a fully synthetic fluorocycline of the tetracycline class developed for the medication for serious infections. The main tetracycline-resistant mechanism is believed to be related to R&D efflux pumps, including AcrAB-TolC/AcrAB pumps. It is still unknown which regulator gene mutations directly promote eravacycline resistance in K. pneumoniae. ramR mutation L58P and F165L reduce the binding affinity of ramR with the RAMA promoter region, thus dissolving the inhibitory effect of ramR on RAMA expression and leading to AcrAB-TolC/AcrAB efflux pumps high expression. These two mutations increase the 2-hydroxy fatty chain proportion of lipid A in K. pneumoniae and reduce the growth rates of mutated strains. When infected with ramR L58P and F165L mutants, a faster organ/blood clearance rate and lower mortality rate are found in mouse models. Elevated pro-inflammatory cytokine responses induced by ramR L58P and F165L mutants in macrophages and human blood. Of all the 500 clinical K. pneumoniae isolates, the susceptible rate was 75%. After being exposed to PAN, 97.6% of strains restored susceptibilities. Expression levels of RAMA, AcrAB, and AcrB exhibited significant differences among the 3 mic groups. We sequenced several main regulator genes of efflux pumps and defined 41 mutation patterns. The eravacycline mic for ramR L58P and ramR F165L mutants increased 64-fold to 4 mg per liter, and this effect was reversible when using PAN. Furthermore, a thigh infection mouse model was utilized to evaluate the resistance level of strains in vivo. The ramR L58P mutation resulted in a complete loss of DNA binding capability. The lethal capabilities of ramR L58P and F165L mutants were significantly lower than those of the WT, also significantly lower initial bacterial loads that were gradually eliminated from all organs. A similar phenomenon exists in peripheral blood, as the ramR L58P and ramR F165L mutants showed a continuous decline in bacterial counts and exhibited significantly lower survived numbers than the WT when incubated with whole blood. In a mouse bloodstream infection model, following bloodstream infections, ramR L58P and F165L mutants exhibited a sharp decrease in blood counts at 0.5 H. To further elucidate the impact of ramR mutant strains on virulence, we investigated whether ramR point mutations could affect genes involved in the lipid A biosynthesis pathway. To further investigate the lipid A structural modification, the mouse spectrum was conducted. The lipid A moiety alteration was confirmed in ramR L58P and ramR F165L mutants, where peaks at M/Z1840 and M/Z2078 were found elevated compared to the WT. To further investigate the weakened blood/serum bactericidal ability of ramR L58P and ramR F165L mutants, we assessed the cytokine levels in whole blood following bacterial infection. As expected, co-culturing with ramR L58P and ramR F165L mutants resulted in a significant elevation of pro-inflammatory cytokine levels in the blood. In summary, we demonstrate that ramR plays a dual role in K. pneumoniae, protecting antimicrobial challenges and highly stimulating the host's innate immune response. Our findings underscore the broader implications of RAMA overexpression, as it leads to increased AcrAB expression and lipid A alterations, culminating in decreased susceptibility to the newly invented last-line drug eravacycline. Our study highlights the critical role of regulation proteins like ramR in regulating survival strategies of K. pneumoniae.